something jogged my mind last year, because last year I've been packing this macro camera in the woods for about 5 years, and it was after this meeting that I thought, well, why am I not pointing it at the back one? Because I'm looking at all sorts of other things, plants, because I'm doing NEPA field work in the Danube National Forest region, and both private and public land, and I just hadn't thought to point that tool at a different subject. So what I see when I go into the woods, why is it not showing up? This is very, very rugged terrain. It's geologically a wedding cake with sandstone cliffs at the top, steep slopes covered in talus, and then limestone and potential caves down low. So you've got a lot of ground to cover. There is typical terrain looking at sandstone from down on a bench where below you would be limestone, and lots of places for a bat to hide. You haven't searched cliff line unless you're looking in holes in the rock. Every single one. I think the lesson from Broke Lake Falls and the Stantec project is, yes, all of the, I was so happy when they found that, because I don't know how many talus piles I have looked at in the last 30 years, and that was the mother lode talus pile, and I didn't, yeah. That was awesome. So I'm definitely searching a needle in a haystack kind of endeavor. The hope is to end up with a bat in hand or in an identifiable distance that you can photograph, or eDNA. Don't throw away clues just based on it not coming to that gold standard. So this is a continuum of certainty, not something that you know right away. I kind of look at it like when you're hunting and you're trying to pattern an animal and you're looking for sign. How do you know where something that's flying through the woods has been? And you can look for guano, you can look for prey remains. In roost structures, there's roost stain, roost splatter, guano, and then recognizing that habitat. So even when the habitat is wet or degraded in some way that doesn't allow it to accumulate guano or roost stain, you could still look at the structure and say, well, if I was a bat, I would definitely hang up here. Acoustics is not my forte, but it definitely belongs on this list. So we've all had the challenge of, here's the guano, we see something that looks like guano but maybe isn't. So the stuff on the left, it's hard to see up here on the screen, but from a distance that looked just like big-eared bat guano. It's got the twist in the middle, it's the right size, but if you look at it with just a little bit of magnification, you can see it's actually probably from a cricket or a snail. So those tubes that have been laid up, and they're really fine because they're eating by rasping, not chewing on the fly. So the remains inside are really fine and indistinguishable, but the way it's deposited, it's like, no, that's not bat poop. And then on the right, you've got wood rat, and that particular dropping, normally they're like Tic Tacs or gel cap size and shape, but it's full of vegetation when you just lightly crush it, so that's not bat guano. When I'm looking at bat guano, the watershed moment last winter was when I go back out in the woods, point the camera at some guano, and I was doing a NEPA project, and for months I've been looking at different areas and mostly finding what was probably big brown epu. And then I went back into Campbell Cave, which was our big find last year for me, and I just spent a couple hours looking at guano, because in the winter the bats aren't present. I mean, there was nobody home last winter, and all of that guano, it comes in a lot of different sizes. So one of the things when we first found that cave, we couldn't believe it was big-eared bats because some of it was really large. I mean, you think about it, Michaela, it's like, this can't possibly be big-eared bats, and yet the eDNA proved it, and then later, you catch 300 in a mist net. That's the gold standard again. So if you're alert to it, the big-eared bats, their prey content's almost all moths. There might be a few stray coleoptera or spiders or something mixed in, but the vast majority of it's moth content, and it's going to be kind of papery, tobacco textured. It looks very different if you zoom in just a little bit. I mean, my daughter can tell the difference with the naked eye. I can't. But with my camera, I mean, it takes 10 seconds to fire up the camera, point it, zoom in. And I started systematically keeping these photos, and when we send in the guano for DNA analysis, I have time and date stamped in my notebook and in my computer full of pictures when and where that particular guano was photographed. So down the road, we really need to do the hard work of proving that this is an acceptable field technique. So moisture is not our friend. For some strange reason, if there's any chemists in the room, I'm kind of curious why we can get hellbender DNA out of rivers and streams, but we can't get bat DNA once the guano gets slightly damp. I'm not a chemist, so but the tough thing is moisture degrades it. So that continuum, some caves are dry enough, whether it's a limestone cave or a sandstone rock shelter, that it lasts almost forever. So you look at it and it just dries out, it loses the mucus coating on the outside, and gets paperier and paperier. And then, like big brown guano and several other bat species that have different prey content, it also loses the mucus coating, but it falls apart more loosely. It's not got that cigarette-like compressed papery texture. But water really quickly makes it difficult. You notice that in this picture here, it's a lot harder to see the difference in texture between those prior slides and this one. It's not as easy to see when it's wet. Plus, moisture is going to make the DNA go away, or at least make it less recoverable for current technology. Really fresh big-eared bat, that picture was taken up there at Broke Leg, and it literally still has creepy crawlies, parasites in the guano. The mucus coating is still intact. So over the course of the year, part of the neat thing about this approach to looking at the landscape and looking up close is you can do it outside of nursery season. So some of this evidence persists year-round. This is in a drainage up in the Clifty Wilderness, and you can still see that it's papery. Oftentimes I'll take either the forceps, or if I'm not going to collect, a fingernail and lightly crush one end of it. And you'll notice in a lot of the pictures, one end is squished. So like back on this, I've played with it. The one that's a close-up, I've kind of flayed it out to try to see inside of it a little bit. Because on the outside, when that mucus coating's intact, it's really hard to tell the difference between whether it's Coleoptera or Lepidoptera inside the pellet. But once you just kind of crush one end, you've got that contrast. And so like this one, I had squished right over the top of the parasite with my thumbnail. And now here is Raffinese big-eared bat guano in a really heavily accumulated rock shelter. It's been a while since we've done any emergence counts on that site, Hood Branch. But it has a remarkable accumulation of guano. So on top, you have an intact pellet this is spring of the year before the bats have returned, so early March. And you can see the degraded material underneath the pellet looks a lot like what's in the pellet, and it's all very uniform. It's all full of nothing but Lepidoptera. And this is the broken down accumulation, and if you really zoom in on its components, it's all bits of moth, both the outside of the body and the wings. So side by side, obviously these cliffs, big-eared bats aren't the only ones using it. And I think we're up to just about every species in Kentucky that's not tree-dwelling has been found at one time or another in the sandstone cliffs. And these feeding shelters, you've got different things side by side. And on the left is probable big-eared bat guano, on the right is probably not, but because of the fine, crunchy content on the degraded one, if I found a more intact pellet of that, I might still sample it because it probably isn't big brown. It looks like it's full of little tiny spiders or caddisfly larvae, that kind of thing. So we've definitely got other critters we're looking out for, like northern long-eareds or small-footeds. So this is a case where I actually got really excited. The people that are working with me as my safety buddy aren't really biologists, and it's been fun. They say, you get so excited when you're in there looking at bat poop. And I saw this little nook, and there was a ceiling pocket. You could see above Ryan's head, that dark shadowy area. It smeared down the side of that rock, right where gravity would cause it to fall, and then bounce onto the floor, were these little tiny bits of bat guano. And I zoom in with the camera, and it's all little tiny diptera and little spiders and stuff. And I'm like, okay, what's this? Unfortunately, we'd been getting stingy with the eDNA supplies, so it went into a pooled sample, but that day I was pretty sure I was going to get small-footed myotis, and it did come up as being in that sample. So once again, one of these days I really want to sit down and make a spreadsheet and determine what level of correlation we have between these 5-second field analysis. This isn't a prey study. This isn't an attempt to say, oh, this is this species. I'm just trying to prioritize which thing gets sent off for $300 worth of lab work. So this was probably the Lebii specimen. So trying to rule out Eptescus fuscus, they seem to be the only thing in the woods that really likes stink bugs. So if you look, I'll bring up a closer slide. And also I've got a big tub here of big brown guano from the nursery colony at the Buddhist temple in Powell County. They swept it up for me and labeled it batshit, but it's actually got visible Chenavia carapaces in it. So they're bright green, bright blue-green. They stick out like a sore thumb. I don't even need the camera to look at that and say, I don't need to put that in a vial and send it to Arizona. So the tricky thing is this continuum. And this whole exercise, I'm looking at thousands of these shelters and caves. Which ones need y'all to go back with a mist net? That's a lot of time and effort to get a team together to go mist net something and find out that, we're not catching anything, or we find maybe one tricolor. How do you prioritize? So the yellow notebook photo is a guano accumulation in the back of an old saltpeter mine that's actually in a sandstone rock shelter. It's breaking down much faster because of the humidity. It's actually behind a waterfall. So even though the back of the shelter is dry, the waterfall is causing a steady degradation of what's on the floor. So the ceiling's dry, the surroundings are dry, but for some reason you can see the salts around the edge of the guano pile. And then the other photo is a close-up of back there in that Hood Branch site where you have this bone dry, perfect preservation. Guano there probably lasts 50 years. Detritivores love guano. When you're zooming in like this, sometimes you'll see the actual slime from snail trails, or you'll find little tiny springtails. So in the upper right, I didn't even see the springtails on that guano until I got home and zoomed in on the picture. The millipedes, you name it, crickets even, will recycle bat guano. But as with the previous picture, in many cases, invertebrate detritivores are going to leave a shadow on the rock where the reprocessed guano kind of holds its shape a little bit. So if you're standing back from a distance and you see some intact guano, and then you see these splotches in the shape of guano where it's all been reprocessed into arthropod pellets, it's probably still guano. I wouldn't put it to the lab, but it helps in the back of my mind as far as contributing to evidence. So how this works out at particular sites is kind of challenging and interesting. Pet Shop is a cave that was found during the 1988-89 Bat Square survey. It's a big spring above the Kentucky River in the Palisades in a fault. I've poked at it from both ends now over the course of the last 30 years. It's a very difficult cave. The bats are in there. When I visited it this summer, so in 2001, I had a report from cavers attending convention. I gave them this cave as a prospect for mapping something virgin. At that point, I didn't know there were bats in it. When we first examined it in the summer of '89, I think my poop delineation skills were not what they are now, and literally the name of the cave, it's covering so many different kinds of poop. At that point, there was actually goat poop in the entrance. So in that picture where I'm standing, this summer, the bats were roosting up and behind my head, but those ledges are covered with pigeon poop, wood rat poop, bobcat, 5 zillion different kinds. But when I go further into the cave, the spatter on the walls that was present at the beginning of the nursery season, so the date on there in June, that week, those bats were in the cave, as reported by the cavers in 2001. So I got a report of a cave full of bats, but it's really, really grim to get to where they are. It's a crevice that's up to 40 feet tall, and at most maybe 8 or 9 inches wide, and you can't just stand up and sideways squeeze through the crevice. You're actually chimneying wherever it's wide enough to go. So interestingly enough, this spatter that was so evident to me on the 7th, when we went back, that spatter was it was in the act of washing off the walls. So that was that ephemeral a sign that the bats were in the cave. You would have had to be there during the nursery season, because now if you went to that cave, those walls would be clean as a whistle. And as they're flying in and out of the cave, they're relieving themselves. And in a dry cave, the walls would show, at Campbell Cave, there's 100 years worth of stain on the walls from the entrance all the way back to where the bats are roosting. But in Pet Shop, that sign is gone 2 weeks later. In this photo, there's what cavers would call a dome, that tall area above my head, and it actually had pigeons roosting with the bats. Because the cave is so difficult to fly in and out of, I think what's happening is that the females are having their young in the cave, and then when the young get a little bit frisky and there's a chance they might try to fly out on their own and die trying to fly in that narrow passageway, the mamas just move out into the entrance, into the twilight zone. And as Sandra and I were walking up on it, I was like, there's bat guano in amongst the pigeon poo. So it's very confusing sometimes. And this is the floor underneath that. There's actually a stream washing away the guano as the bats are pooping. So it's very transient in that particular cave. And it's only because I'm looking at the whole cave environment that I can say, I just got lucky here. We're here at the right time. The habitat was all great and suitable, and you know, we've been looking at it since 1989 because we knew it was a really awesome looking spot. Prey remains, add some context. I used to think that you could tell a lot by what moth species, because there are some species that there's a temporal dimension to when they're emerging. In general, your underwing moths are going to be midsummer to fall, and then there's a lot of others. I just thought it was cool that the herald moth was underneath the big-eared bat roost, and you could see those two little holes on the midline up near the stripes. That's a pair of tooth marks. So John challenged me years ago. He's like, everything eats moths. How do you know that this was eaten by a bat? I was like, birds don't have teeth. So you've got that gap in the palate and the two big canines, and that's usually, if you hold up a mothwing to sunlight, you'll either see that, or in some cases, some other sign that a different kind of animal probably preyed on that. So these are examples of mothwings under a phoebe nest. So you definitely need to be looking at the ceiling. If you see a phoebe nest and you see a pile of mothwings, notice that the bases of the wings have little tufts of the body fuzz from the moth on them where they're plucking. And unfortunately, you can't see it on this screen up here, but on one of these, there's actually a triangular beak print. If you look at it blown up on the computer screen, you can actually literally see where the phoebe's beak took scales off the mothwing. So it's kind of strange. So these are crunchy Coleoptera. The green is the Chenavia, the native stink bugs that big browns love so much. I'm not going to spend to process guano from the shelter where those are the prey remains I'm seeing, because it's probably big browns. The other context thing is I think big browns, they're the big bruiser of the bat world kind of, and they don't seem to care how sheltered they are. They seem to roost in more open settings. So in a sandstone site, they may be wide open to owl predation. And I don't think anybody else is out there daring the owls to come get them. As we know, there's a lot of moth defense mechanisms evolved to protect themselves from bats, and certain species just don't show up as prey remains in a bat shelter. The underwings are highly evolved to avoid predation by bats, but apparently they taste really good because they are definitely a bat favorite. Some of these tiger moths, I've never seen them eaten by bats or birds. So they obviously have a chemical defense. So what these structures, whether it's a sandstone shelter or a limestone cave, they need to provide a dry ceiling area with a warm air trap ideally. Dark zone, I think they just don't want to be seen by predators. So dark-ish. Obviously, big-eared bats are really tolerant of being in the twilight zone. And I'm starting to think that background water noise is a feature, not a bug. So in acoustics, we've tried using the Bad Elf acoustic monitors on a lot of these shelters, and probably 60% or more of the caves and shelters we're trying to study, there's too much water noise around, too much environmental noise for acoustics to work well in that place. So if you were an owl listening for squeaking and it's hiding underneath the sound of a drippy waterfall, that might actually be a defense mechanism. When we first started doing Cliffline for John, we're a bunch of cavers, so we were using caver-type language in many cases. And then I started rock climbing, and some rock climbing terminology bled into my notes because you're trying to describe different kinds of rock features. What makes that little area that's a shelter for a mama bat. I think it helps if we all try to sort of use the same language when we're taking notes. Crevices are probably the #1 structure. So Pet Shop Cave is a crevice a half-mile long. There are sandstone shelter caves that are probably just as long. Getting my body into them is probably not possible, so we're going to have to come up with other ways to further investigate those sites, because a human body just isn't going to go there. So domes are one of the places where caver language we've parted ways a little bit. I think for biologists we're using it largely as any high ceiling that forms a warm air tract. Whereas for cavers, a dome has a much more specific meaning as far as it's kind of roundish and usually formed by dripping from above. Charlie, correct me if I'm wrong. Particular barbism that has persisted is the igloo or phone booth structure. I use that as a general catch-all for limestone and sandstone sites where you have a constricted crawlway entrance that opens up into a room of some substance, and quite a few of those have been shoulder season sites, and I guess you could say in a way that Placotis Pit is that structure, because you've got a fairly constricted entrance that opens up in the back and it's not very extensive. Talus, cavers call it breakdown. You probably recognize the site on the right, James. The one on the left, literally, thousands of holes like this if I shove myself into, and you got the mother load. But it's potential bat habitat. It's dark, it provides the warm ceiling, and that one has water dripping across the floor, making a little noise for predators. Where geology and rock climber language has leaked in are these huecos, would be the big holes in the cliffs. And these are actually usually only in sandstone. So there's just something cavers would call solution pockets in a cave. And that would be a completely different mechanism of formation. So huecos and Tafoni, the little tiny pockets. If you scroll through rock climbing online guides and look at, they do what's called spray, and you can find bat reports in that. Almost got my finger bitten. They're hanging from this little finger pocket, and they stick their hand in there, and there's a bat in it. and so we know that bats are using it. We just don't know what species, because I'm not climbing anymore. In reality, all those terms kind of get combined in many cases. So you'll have an amphitheater that'll have a crevice in the back, or you'll have a crevice that turns into a phone booth room, or an amphitheater where there's talus across the front of it, which makes a cave-like structure. You can mix and match the terms, but it's a descriptive terminology shorthand that I think helps us describe structures that might be beneficial to the bat. So, these pictures are only two-dimensional, and sometimes we only take an entrance picture, or sometimes we forget to get some scale in the picture. And if in your notes you have dimensions written down and a few of these shorthand words to describe the structure, I think that really helps us going forward. So this is just a small sample of what GPS tracks look like when you are running cliffline. There's also limestone down low in this drainage. You could probably see by the squiggles on the map where we started finding bat sign. That area had been covered intensely in 1996 for a trail corridor by both Dan Dorson and I, and 90% of that area we found no bat sign. Found dozens of structurally suitable shelters. So looking for that guano, looking for those prey remains, even though they're suing everything. We don't know why, but it was really exciting to get into those areas where you started finding things. And that was one of the first places I started applying the macro camera. And I immediately knew I was starting to find big-eared bat guano. And then where those nests of tracks are, that would be where the sites that we found that had Virginia big-eared bats in them were. But you can see it's very labor-intensive, but it's only, you know, a couple people. You can do it with one biologist and one safety buddy. Basically, I think what we're using this field technique for is to triage these miles and miles of miles and miles. You are trying to decide what gets future inquiry, what gets a missed net visit, what gets the resource expenditure of sending in guano for identification. And I think you want the bat sign to be there. Is the root structure there? And then history. So James, you knew that you'd found some covey in the area where you guys were going to be mist netting. So you were probably a little excited going into it, hoping you were going to catch something. These things don't come out of the blue, very rarely. We get lucky every now and then, like at Campbell, but in most cases, like Pet Shop, we have been picking at these sites for 30 years. It's just persistence. Sheer just sweat and grind going through roto hell. So anyway, you guys got questions? I've got a tub of 2 different kinds of guano. The big tub is big brown. I've got my macro camera, and I've got a vial of big-eared bat guano from Campbell Cave, so that if people want to kind of get hands-on and see the differences that I'm looking at, you're welcome to come play with bat poop with me.
We've got some good if I have it.
Barb, one of the things you were talking about, and I'm by no way a DNA expert, but I have been dabbling in eDNA.
You've gotten in the room with them.
Recently.
I haven't.
But one of the things, too, is you was talking about how the water was breaking down the guano. It may just require a different method of analyzing that guano, because when we were doing the Roadrut Pond stuff, the way that they were doing it in northern Arizona was not sensitive enough to pick out the bat DNA.
Right.
So then we did it a different way. I can't remember the terminology for it, but I can put you in contact with Nathan, our eDNA expert. But
Can I ask you a question back? So this awareness thing, when you talk about those Roadrut ponds, you can see in the mornings those moth wings floating on a Roadrut pond. And if you're looking at them, hold them up, look for tooth marks. And then that's where you set your mist net, isn't it?
yeah,
Or,
well,
or your camera.
We're hoping to follow up and do some eDNA.
Which is awesome.
Of some of those road ruts where we find sign this coming year, hopefully.
Cool.
And but the interesting thing is too, is when you have, like in the road ruts, if you've got a lot of amphibian activity in that road rut, the DNA from all of that just covers up the bat DNA. So
Wow.
you may have the same thing going on a little bit with some of your guano, because the insect parts is covering up what little bat guano or bat DNA is in there.
Plus, you know, that's probably water soluble because it's coming from the intestinal lining of the bat.
Yeah.
So
The other thing was too is have you been back to the Talus shelter? I think it was in Walker Branch when we were doing the pipeline stuff back in the early 90s.
I need
That
to.
was a Talus cave.
That was a Talus cave.
I remember you getting really mad at Hal for dislodging the big boulder above you. That was really funny, by the way.
Which is how I've had shoulder surgery 30 years later.
Good talk.
Okay, thanks guys. Anybody else got questions? Young people? Yeah.
I was going to ask how you can tell the difference between bat stain and, since these rock shelters, talus areas are close to the surface,
Soil.
root stain, soil stain, different properties of the rock, and that throws me.
Right. So I don't think for colonies, our bats really, I don't often find them roosting in. If you look on the underside of the talus, there's often little spider webs of rootlets and things. And I flushed a few out of sites like that when I was near something bigger. I have a few rules of thumb, kind of, that people who've been in the woods with me will have heard before. The raccoon rule, and then if you're flushing bats out of stupid places, there's something bigger nearby. So when you're flushing bats out of a hole in a root wad, if it's a cave species like a bigger bat or something, you need to really look hard within a quarter mile of that area. So as far as the soil itself, though, I don't think the bats really like that substrate, and physically there's an oily texture to the staining. So when you actually zoom in on it, the dirt's going to look kind of fluffy or unattached, and then the guano stain is going to have kind of a greasy nature to it. And if the bats have been there in the last year or two, there may actually be hair in it. So one thing that doesn't show up on this screen is almost all of this. I think part of big-eared bats being so social, their guano seems to have a lot more hair in it than than other species. So like if I'm looking at a big brown's guano, you find some hair, but you don't find nearly as much. I mean, even this is from a nursery colony of big browns. And compared to what's in the covey guano, there's hardly any hair. And you think they're up there grooming each other. And so that stain on the wall at a place like Campbell Cave, if you zoom in on it, it's not just bat poop. There's actually animal hair in it. Does that make sense? It's hard to understand how quickly this tool changes things, because this thing's literally in my pocket most of the day. It's waterproof, it's fairly shock resistant, and I'm zooming in on things, and it takes maybe 5 to 10 seconds. And I snap a picture of my GPS for an index picture, so I know.